Hey, welcome to the Biochemistry Academy. Imagine you have a special key that can only open one specific door. Now, this door represents a chemical reaction, and the key is like an enzyme.
Yeah, boy.
Just as key fits perfectly into its lock, enzymes are highly specific proteins that fit perfectly with their substrate to facilitate or speed up biochemical reactions. Now, in this video, we will focus and understand what enzymes are, understanding the characteristics of enzyme, and we will also talk about the classification of enzymes. Now, the first question is, what are enzymes? Enzymes are specialized proteins which are produced by living cells to speed up or to catalyze biochemical reactions. Now, the molecule in which an enzyme acts on is called a substrate, and this substrate is finally converted into a product. And we should know that the study of these enzymes is called enzymology. Now, enzymes are known to catalyze more than 5,000 reactions taking place in our cell. Remember, we said that these enzymes are specialized protein, meaning that they are made up of protein, although there are some RNA molecules that can act as enzyme. For example, ribosome. Now, enzymes are highly specific for their substrate, and they accelerate this specific reaction without being consumed in the process. Now, let us understand it with this simple reaction. This is the substrate in which an enzyme acts on. So by the action of this enzyme E, it forms an enzyme substrate complex, which is an intermediate. Now, this is further converted into products. And if you see, the enzyme has gone back to its original form. It is no longer joined together with the substrate. But if you can see, the substrate is being converted into a product. And that is what we mean by enzyme. They only speed up the biochemical reaction without being consumed. So now let us talk about the characteristics of these enzymes. Number one, enzymes are highly specialized proteins, except for ribosome, which we mentioned earlier. So because you say that enzymes are specialized protein, meaning that they follow the physical and the chemical properties of protein. They are water soluble and they are heat labile. Enzymes can be precipitated by protein precipitating reagents like ammonium sulfate. And next, enzymes act under mild condition of temperature, like in the human body with a temperature of 37 degrees Celsius and in a pH of 7.4. Another characteristics of enzyme is that they contain 16% of nitrogen. Then enzyme also undergoes denaturation and regeneration. We will talk about it more in our coming videos. So next, let us understand the classification of enzymes. Enzymes are classified based on the International Union of Biochemistry and Molecular Biology, which is abbreviated as IUBMB. Now, in this IUBMB classification, each enzyme has a code. It has a code number, and this code number is called Enzyme Commission number or EC number. The EC number has 4 digits, and we are going to use the enzyme hexokinase, an enzyme that is involved in glycolysis or glucose metabolism. The EC number of hexokinase, it is 2711. Now, the first digit represents the class, and in this case, it is a second class, which is class 2, and which is transferase. Then the second digit represents a subclass, and there are 7 subclasses, but for hexokinase, it is in the 7th subclass, which is phosphotransferase. The third digit represents the first subclass, where an alcoholic group is the recipient of the phosphate group, and the fourth digit represents the individual enzyme or the first enzyme, and in this case, it is hexophosphotransferase. Now, in recent studies, there are 7 major classes of enzyme, which are EC1, EC2, EC3, EC4, EC5, EC6, and EC7. And the EC1 is called oxidoreductase. EC2 is transferases. EC3 is hydrolysis. EC4 is lyase. EC5 is isomerase. EC6 is ligase. EC7 is translocase. And remember, enzyme always end with the suffix ase, apart from the RNA molecule, which is ribosine. One can be creative to create a mnemonic, like for example 1, 2, and triple H. 1 can represent oxidoreductase. 2, the number 2 can represent transferase. Then triple H, if you remember the wrestler, and this H represents hydrolase for the first 3 EC numbers. Then for the remaining 4 EC numbers, we can use the mnemonic LIT. We can say that the topic of enzyme is LIT. You can use it to remember, but you can create your own mnemonic. So let us look at the first classification of enzyme. And we say EC1 is oxidoreductase. Now, they are enzymes that catalyze oxidation and reduction reaction in the cell. And if you remember in one of our videos, we said that oxidation, it is the removal or the loss of hydrogen or the loss of electron, while reduction, it is the gain of hydrogen or the gain of electron. Now, an example of enzyme in this first classification are alcohol dehydrogenase and cytochrome oxidase. Now, let us look at the second classification, which is EC2, which is transferases. If we check, as the name implies, these are enzymes that transfer a functional group from one molecule to another. Now, example of enzyme in this classification are the hexokinase. Remember when we were talking about EC numbers, the 4 digits, and we said that too represent class 2, right? So hexokinase is an example of transferase. Then let us look at the third classification, which is hydrolase. Now, these are enzymes that catalyze the hydrolysis of various bonds. It involves the breaking of bonds by the action of water, and an example of enzyme are lipase and amylase. Then let us look at the fourth classification of enzyme, which is EC4, which is lyase. Now, these are enzymes that cleave or break bonds other than hydrolysis or oxidation. Now, without the addition of water, they break bonds, leaving behind double bonds. The next classification is C5, which is isomerase. Now, these are enzymes that catalyze isomerization reaction in a single molecule. Now, the next classification of enzyme is C6, which are ligase. And these are enzymes that joins two molecules by a covalent bond. An example of enzyme in this classification are acetyl-CoA carboxylase and glutamine synthetase. Then let's look at the next classification of enzyme, which was added in the year 2018, EC7, which is translocase. Now, these are enzymes that catalyze the movement of ions or molecules across the membrane. An example of such enzyme is ATP-ADP translocase and orthinin translocase. In our next video, we'll continue this serial course on enzyme. In our next video, we'll talk about cofactors and coenzyme. Please don't forget to like, share, and subscribe to the Biochemistry Academy for all biochemistry courses. Arigato. See you in our next video.